We are going to move forward and discuss a really interesting technique that is used in cell biology, centrifugation. This allows us now to go into the cell and start asking questions about cell physiology, the relationship of organelles, biosynthesis of organelles, movement in and out of organelles, protein targeting to the organelles. Lots of questions. In cell and molecular, we want to isolate a part of the system and then deconstruct that. Okay, so that's been at least what I've tried to emphasize as an overall landscape for research in cell and molecular is you find a part of a system and then break it down. So centrifugation actually allows us to isolate parts of cells for further investigation. Let's see here. Yay! Okay, so I'm using the purple color today. Okay, so just stepping back, so kind of, in concept maps, I like to look at the big landscape first. So this is a slide called General Steps in Biochemical Separation. And this may be more in a protein level, but at least you get the idea of some of the things we're going to try to do here in the next lectures. We obviously need a natural source for our cells and proteins, so that probably is cell lines, could be primary cells, again, could be disease, cancer cells, depends. So we need to extract those cells, we need to break them open, we need to start separation by centrifugation. A lot of times after we break open cells, we get something, a crude extract. We can freeze that down, save it for later, or we can put many cells together and concentrate an extract. Remember, some of the techniques we use require a lot of samples, so sometimes just one cell is not enough. We're talking multiple cells, and so we're assuming that those cells are much alike. But just in the back of your mind, remember that things like cancer and tumor cells can be different. So, but this is just a kind of a map here. You have to design and have a map, a road map, a concept map, an outline for isolation and purification of the organelle, the protein. And we'll talk about protein fractionation coming up, how we actually separate proteins. We will certainly in this lecture discuss centrifugation. We will talk about chromatography techniques and then electrophoretic or electrophoresis methods here. And so we get the purified product. We're hoping that it's high yield. In other words, we get a lot of it from a little sample. When you're doing biopsies and things, sometimes you don't have a lot of sample. We want it to be pure protein or pure organelle, pure nucleus, right? Nothing, we don't want ER or Golgi. And then we want to set this process up to maintain the biological activity. Where we would like to go is once we have an organelle, we would love to figure out a way to mimic that organelle's function and activity in a cell. It's called cell-free system, and we will touch on that coming up as well. So that's kind of an overview, the general steps in a separation, and we'll see kind of these things again. And so separation of macromolecules, We use the techniques, chromatography. We can precipitate materials out of a solution. So probably in your basic chemistry, you did some type of separation techniques where chemicals and things could become pelleted out of a liquid here, right? I mean, if you put too much sugar in a glass of tea, a lot of the sugar falls to the bottom. That's it. It's precipitated out because it's the iced tea is saturated, right? So there's a point where proteins and whatnot precipitate out. You just can't add any more to a liquid. Electrophoresis allows us to separate materials from each other, and we'll talk about that. And then we're talking about centrifugation, and the highest level of centrifugation is called ultracentrifugation. So this is the fastest, the most precise type of centrifugation. And this just shows you the typical cartoon of an animal cell here, the different organelles, and we will cover all these at some point in this course. And it just kind of shows you the volume. I mean, almost half is cytoplasm. So right away, a big separation is organelles and cytoplasm. Can we separate out, kind of like in Jello, can we separate out the grapes from the Jello? I don't know if y'all have ever had Jello with grapes inside, but it's kind of maybe an old dish from the old days, if you like grapes. Okay. So we have to separate out. So you can think of the separation as a big step first, and then the materials can be further worked on. So you kind of split it apart, right? Bread, you got the crust and the center, and then you can work further down if you can separate it. Organelles, cells, materials have a certain density. Density is a physical biochemical term, and that is discussed in terms of grams of the material per centimeter squared in its environment or in a liquid or in some type of media, and you get a density. In other words, when a cell reaches the layer in the material that's the same density, it will not separate further. So we use density as a way to separate because there are differences in bacteria, in mammalian cells, in organelles, proteins, DNA, RNA. So right away, because of density differences, you can see theoretically if you have a bacterial infection in a blood, you had that blood sample, you could use density to separate the bacteria, separate the red and white blood cells. We could take those blood cells and pop open the organelles. From the organelles, we could get the proteins. And of course, in the cells, we have the nucleus. So we have the nucleus here, and then the cytoplasm with its organelles there. Proteins can be in the organelles, and of course, there's histone proteins. Okay, that's my arrowhead associated with DNA. And again, we got densities. So right away, we're able to take advantage of the differences in the density of the material that we're trying to separate. So this is a cartoon showing a rotor. A rotor contains samples, and this little circular curlicue is rotation. And then it shows the different densities, and you can see kind of the size, and here's kind of the smallest density. And so it's saying that applying cells to a centrifugation will separate organelles, the nucleus, some of the other ER membranes, Golgi, mitochondria, and then any kind of free proteins. So it's a device, not a cheap device, for separating particles, organelles, proteins from a solution according to size, shape, density, the viscosity of the media, and how fast we spin it. So viscosity means how thick it is, right? So it's the same thing if you've ever had to use the liquid plumber and you poured it into your stopped up sink, it goes to the bottom. That means it's a very dense, and it's much denser than water. Okay, if it was not denser than water, it would stay at the top. It is dense, it drops below the water. So you think about that concept of density here. So in a solution, particles whose density is higher than that of the solvent sink. And that could be water, could be glucose, glycerol. It depends on what you're separating the materials in. Particles that are heavier will sediment to the bottom, towards the bottom, and particles that were lighter will float on the top, right? Oil floats on the top of the water, okay? A stone sinks. The greater the difference in density, then, in other words, if they're very dense, they're going to move fairly fast. If there's no difference, and that's called isopycnic, and that is a whole type of centrifugation. It's a flavor called isopycnic centrifugation. If there's no difference, then everything has reached its equilibrium and they stay steady or stay in equilibrium. So iso means frozen or the same. And so if you remember isotonic, right? No net flow of water in and out of a cell. So isopycnic, we're talking about density. All materials have reached its density inside whatever solvent you've placed them in. Again, in centrifugation, we use glucose, sucrose, all sorts of kind of thick solvents, and we'll talk about that. In other words, we don't use water necessarily. We could, but most everything would be denser than water. So to take advantage of small differences, gravity can be replaced with centrifugation. So yeah, in liquid plumber, we just pour it in. So gravity pulls it down because it's denser. If we apply a centrifugal force, we can separate things that are very close in density, completely separated. And so that's why we're doing centrifugation, is we need to apply a centrifugal force. Gravity will not work, okay, because a lot of the materials, if you remember back to the chart, there was a lot of overlap in some of these materials in density. So centrifugation, apply a centrifugal force, allows us to separate the components of a cell. A centrifuge will separate micromolecules or particles, cells, subcellular components, proteins, nucleic acids, and these components would be organelles here. I don't know why they say subcellular. Should be organelles. Okay, there may be like ribosomes, okay, could be considered a subcellular component that is not an organelle. So maybe that's why. So it's including the ribosomes, okay, ribosomes, and organelles. Okay, it's kind of hard to draw here. Organelles. Wow, look at that G, organelles. And I can't spell, but that's okay. You get the message. Organelles, looks like arginelles. That's an O there. Okay, and ribosomes. Okay, these are dense particles here. Ribosomes. And again, the basis is size, shape, and density. and we use the density differences and the solvent or the media that they're put into. And then we apply an induced G-force through centrifugal action, spinning in a circular motion here. So that is, in a probably not a nutshell, but that is centrifugation. Centrifugation is a tool of subcellular fractionation. So subcellular means below the cell. Fractionation, we have to optimize all the process to get a clean, pure, quick separation. That's what we want to do. If we get to the protein level, there's a big hydrophobic versus hydrophilic proteins. Okay, so that can be an issue to separate those together, and there's some tricks to maintain membrane structure in a separation. We would like the nucleus to be intact at first, and then we can break the nucleus once it's pure, mitochondria, other organelles. So here's a cell suspension. We shear cells. There's a little kind of a little drill bit kind of that spins these cells and breaks them open, so they get popped open. This is one way. There are other ways to pop open cells. And so now all these cytoplasmic contents fall out. Homogenization leaves most of the organelles intact, so that's a key component there. Okay, if we want to study mitochondria and mitochondria are destroyed by this process, then it would not be a valid process. So we can centrifuge the homogenate and get a pellet that contains all the organelles. We can then permeabilize, add buffer vortex, and we can separate cytoplasmic or cytosolic proteins, and we can collect the supernatant from, and by the way, we do a lot of centrifugation at 4 degrees. When in doubt, chill out. That maintains the structure and keeps proteins from being broken apart. And we can resuspend this pellet, these cytoplasmic proteins, okay? And eventually we can get membrane proteins out of these total cytosolics. So there's hydrophilic proteins, hydrophobic, and the hydrophobic will be in the membrane, and we can isolate membrane proteins here. So this is at the protein level, but we probably start first with isolating the organelles, but you can go further, and we will talk about protein fractionation. This is some words here, hopefully discussing what I've done, and something we hadn't talked about. Revolutions per minute, or G-force, is the measure of the amount of centrifugal force applied. And again, size, shape, density, the amount of solids present, the density difference, very key between what we're trying to separate and the solvent, the liquid it's in, and how viscous, how thick is the liquid. Types of centrifugation, okay? There are types that rely on a very high maximum speed to sediment materials. Sometimes it's so fast that we have to do it in a vacuum because of the heat generated. Okay, if you didn't, you would heat up the centrifuge and it would probably blow up. We also have temperature controlled refrigeration of our centrifuge to keep the temperature cool to maintain the structure integrity of the material. And we have to pay attention to the volume and capacity of the special tubes. These are not just paper cup tubes. These are hard plastic that can withstand obviously high levels of force. And depending on the force levels depends on the type of centrifuge tubes and their makeup. So you have to order special tubes from the makers here. The easiest that we use is called a microfuge. And I don't know if you can tell, but this is about, size of a quarter or 50-cent piece here. Okay. And it holds about 1.5 mils of material. So a very small amount, but that may be what you have for a sample here. And then you put it into these little black holes. This is a rotor, and these holes are where you put your tubes, and they need to be balanced. So if I put a tube here, then I have to put a tube there in the same amount of liquid to balance it. If you put the tube next to it, it will be out of balance and could destroy the centrifuge, and people will get cranky. There are large capacity centrifuges that are big preparative. In other words, if there's a lot of cells, you're trying to isolate some monoclonals from liters full of hybridomas, then you might kind of spin the cell in a rotor bottle. This is a 1550, and there's actually 250. There's a liter. There's 5 liter. So they can be really big containers, and the rotors used are selected by what they can hold and withstand in our centrifuge here. This is a steel walled centrifuge, and inside is a vacuum container. Part of that steel protects if the centrifuge fails, i.e. the rotor comes out here, it's going to be spinning at maybe 50 or 100,000 G. I assure you that rotor will go through walls. Okay, so that's called a catastrophic failure. Just look on the internet and look for examples where centrifuges have failed. You're saying, how do you stop failure? Well, the rotors have a life, and you have to keep records, and you can eventually they get derated. That means you can't spin them as fast. It's kind of like an old car. You don't want to try to top it out after 15 years of driving. Probably couldn't handle 100 miles an hour, okay? But a brand new car probably could. So it's called derating the rotors. This can be 100,000, 50,000, and these can be 2,000 or 3,000 or more in cost, just for you to get an idea. Microfuge cost 1,500, maybe 1,000. So just kind of give you some cost here. There are high speed, and these are 100,000. That's the ultra centrifuge. They will separate nucleus, mitochondria, proteins, large organelles, debris. And then finally, 600,000 is the ultra here. And these will get ribosomes and the very small dense particles here. Because of the friction, we have to do it in a vacuum that's evacuated and has to be cooled. And here's an example of an ultra there. So depending on if we were looking for viruses, we would be in this range. If there's a new virus in a patient sample, we would have to be in that range because it's so small. Here's centrifuge rotors showing the angles and how there's separation. Pellets, swinging buckets allow you to have a pellet almost at the bottom. This gives you a slanted pellet. The slant meant for short time runs, very quick, most popular, but longer separation better, you may require a swinging bucket. And this makes it easier to draw out the liquid. Here's the liquid. That's the supernate. This is the pellet. Okay, make sure you got those terms without disturbing the pellet, right? So Just again, it's empirical, depends on what you're doing. Okay, so you develop a roadmap. You just don't find a rotor and stick something in there. You got to know what the rotor does, how fast, what are you trying to separate. Here's just some more. There's also vertical tube rotors. And here's the swinging bucket, and it's just showing materials getting separated. And this is showing, here's 5 materials at different density. This would be high density. This would be lower density here. Okay, so you can see right away we get a separation by density from low to high. So think about that, when we're separating materials here. Okay, so that leads us into cell lysis. And what I'm going to do is stop now, let you reflect on just kind of the overview of centrifugation. For cell biology, we're usually either isolating proteins or we're isolating organelles. In clinical situations, you may be isolating viruses, bacteria, fungus, parasitic eggs, and materials. So it depends on the clinical versus the basic science, what people are doing. So there's clinical labs and there's basic science research labs, right? And right away, you could imagine that a cancer cell would have a different density profile than maybe a wild type normal cell. So that might be a way to start, is just, why is the cancer cell different in structure, shape, content? Okay, how did that happen? Where do we go from there? Can we reverse that? Can we kill the cell? You have to understand the process and the cell before we can then move to that next step. So think about centrifugation and all the kind of possible applications that it can use. And certainly in biochemistry, you will get into centrifugation. So this will help you out when you hit that. So come back and we'll talk about cell lysis. And we will see you very soon.